Hello, this is part 1, and I'm going to be answering from question 1 to question 4. This is for Pearson Edexcel International A-Level, January 2025. So this is a very recent paper. And this is for Unit 1, which is Molecules, Diet, Transport, and Health. Okay, so question 1. The polysaccharides and proteins that we eat are digested. The products of digestion are then transported in the blood plasma as a solution in water. Which of the following statements about water are correct? Notice how you can technically just read A and then answer the question, but they usually give you one or two sentences to read. Sometimes these sentences are important to read, and other times they're a waste of time, honestly. Okay, so 1. A single molecule of water is composed of an oxygen atom joined to two hydrogen atoms by hydrogen bonds. Okay, now this part is incorrect. Okay, so water is H2O. So the thing that's wrong about this is this part by hydrogen bonds. So this by hydrogen bonds, they're talking about this part. This part over here, which joins the oxygen to the hydrogen in the water molecule itself, is called a polar covalent bond. So this bond is called polar covalent. This part over here is called the hydrogen bonds. This is between two different water molecules, okay? So over here they're talking about a single molecule and the hydrogen bonds, which is wrong. The hydrogen bonds are created between two different molecules, not a single molecule. So the second part, water is a solvent. Correct, water is a solvent because water is polar, meaning it has a partial positive and negative charge. The partial negative is on the oxygen, whereas the partial positive is on the hydrogen. So water is a polar solvent, okay? The oxygen in a water molecule has a small negative charge, and the hydrogen atoms have a small positive charge. That's correct. So therefore, our answer is C, where only 2 and 3 are correct. Okay. The table shows some information about the structure of amylose and amylopectin. Put one cross in the appropriate box in each row to show whether the structure is present in these molecules. Okay, so amylose is unbranched and it has alpha glucose. Alpha glucose, remember, you need to know the difference between alpha glucose and beta glucose. So alpha glucose, the hydrogen is above the hydroxyl group, okay? So amylose is unbranched alpha glucose coiled, which makes it compact and meaning it's good for storage, okay? Whereas amylopectin is branched chain, and the side branches allow enzymes to break glycosidic bonds easily, meaning that glucose can be released more easily, therefore releasing energy faster, and it's also alpha glucose. So both amylose and amylopectin contain glycosidic bonds, and they contain 1,4-alpha bonds because they're both alpha glucose. Now, branched side chains are only present in amylopectin. Remember, while they're branched, for the enzymes to break glycosidic bonds more easily to release energy faster. A way to memorize this is by creating flashcards. So when I did my A-levels, I was doing flashcards based on every question that I get from my past papers. Okay, the diagram shows parts of two amino acids. Complete the diagram to show a peptide bond joining these two amino acids. Okay, so an amino acid is formed by the removal of water. This reaction is called a condensation reaction because we are removing water, okay? So an OH group is removed from the carboxyl group, whereas a hydrogen is removed from the amine group. Oh, that's a yellow color here, from the amine group. Okay, so the side with the amine is going to have the carbonyl group over here, okay? And then the side with the hydroxyl over here is going to have the amine group over here. This is our peptide bond. all these marks till now, there is nothing difficult and you should be getting them. So you should be aiming to get these 5 marks because these questions are very easy. Okay, cancer can develop in many parts of the body. State what is meant by the term cancer. So cancer is when cells are dividing out of control. Mitosis is occurring without stopping, okay? That's basically the same point as saying cells are dividing out of control. And this is caused by a mutation in DNA base sequence. You're doing A level now, so it's really important to be specific. So you can't say it's caused by a mutation in DNA. No, you have to say caused in the mutation in DNA base sequence. You have to be very specific. Okay. In a study, scientists determined the number of deaths caused by 5 types of cancer in one country. The scientists then analyzed the number of deaths caused by these cancers in 3 ethnic groups. The graph shows the results of this study. So we have our key over here: lungs and bronchus, colon and rectum, breast, pancreas, prostate. So have a look at these values because these values are all going to be needed for the coming questions. Okay, notice here we have ethnic group 1, ethnic group 2. Notice how for ethnic group 1 compared to ethnic group 2, the prostate cancer in ethnic group 2 is higher compared to ethnic group 1, whereas over here it's the second highest and over here it's the third highest, okay? So compare and contrast the numbers of deaths caused by these cancers in ethnic groups 1 and ethnic groups 2. So if you remember the graph that I just showed you, the highest cause of death is lung and bronchus in both. So the blue color represents the similarities, okay? The second point, the lowest cause of death is pancreas in both. And then the difference is more people died of cancer in group 2. Okay, which is the percentage difference between the number of deaths caused by lung and bronchus cancer in ethnic group 1 and ethnic group 3? Use the formula. So we have percentage difference equals the difference between the values, meaning that you're going to substitute, divided by the mean of values. Remember the mean, you add and divide. You add and divide for the mean, okay? Times 100 for a percentage. So if you go back to the graph that I just showed you, you have 47 minus 18, okay? And then you're going to add the 47 together, divide by 2, because they're 2 different values from 2 different ethnic groups, times 100, which gives you 89.23%. Now, calculate how many more males died of prostate cancer in ethnic group 2 than in ethnic group 1. So ethnic group 1 gave us 18, which I rounded to 20, and ethnic group 2 gave us 38, which I rounded to 40. Now, since this is just a 1-mark question, you don't need to calculate or write down anything. However, I always recommend you write it down. So you can see that 40 divided by 2 gives us 2, so that means it's twice as many, or you can simply write 2x. Okay, so comment, so keyword on comment, okay? Comment on how useful it is to determine the number of deaths from these causes of cancer in the all ethnic groups part of this graph. So focus on comment and all ethnic groups part of this graph. So number 1, it's not very useful as cancer cause different number of deaths in different ethnic groups. If you go back to the graph, all numbers of deaths are different, okay? Number 2, however, it could be useful as you can get an idea of the effect of the different cancers overall. Now, these types of questions are always going to come in all your A levels and all your exams. It just wants to see the way you're thinking and the way that you're analyzing data and putting scientific cases in real life scenarios. Okay, question number 3. Bears hibernate during the winter months, so are inactive for long periods of time. The graph shows 2 bears hibernating. Okay, we get it. Hibernating bears do not develop deep vein thrombosis, DVT. Deep vein thrombosis is a blood clot that forms within deep vein, often in the leg. In humans, short-term periods of inactivity, such as sitting in an airplane, increases the risk of DVT. This is due to reduced blood flow in veins, causing damage to the walls of the veins. So which molecules released from the platelets results in blood clotting? Well, it's going to be thromboplastin. Remember, when we have a damaged blood cell, this will release thromboplastin. Thromboplastin will combine with the calcium ions present in blood, which will activate the reaction. They will act as a catalyst for prothrombin to turn into thrombin. Now, thrombin is going to act as the catalyst in the enzyme for fibrinogen and turn fibrinogen into fibrin. Fibrin will form a network like this of fibers that traps blood clots and platelets. It will trap them like this. And then the platelet will form a scab. So this is our scab. And they're trapped in the fibrin network. Okay, so we said it's thromboplastin. Now, which pair of molecules are enzymes in the active form? Well, they're going to have to be thromboplastin and thrombin. So the answer is C. Now, which molecule found in the blood is insoluble? Well, it's going to have to be fibrin, right? Because this is the mesh that stops us from bleeding out. So it has to be insoluble. So it's fibrin. Now the rest over here are insoluble. Okay. Pulmonary embolism can be caused by DVT. Remember DVT, deep vein thrombosis, when the blood clot travels through the circulatory system and blocks the pulmonary artery. Okay, the pulmonary artery is the artery that takes deoxygenated blood away from the heart and it brings it to the lungs to get oxygenated. Okay, so symptoms of pulmonary embolism include difficulty in breathing and a rapid heart rate. Suggest why pulmonary embolism can result in these symptoms. Well, number one, the blood clot in the pulmonary artery prevents deoxygenated blood from reaching the lungs. So, blood will not become oxygenated. Therefore, there will not be enough oxygen supplied to the cells. So the heart is going to have to keep pumping more, okay, to get the most amount of oxygen and most amount of blood around the heart, since the blood is going to already have a less or a lower concentration of oxygen. I find this question really interesting. And over here you can see the blood clot in the pulmonary artery. Okay. A study of bears in Sweden discovered that these bears produced less HSP47 protein when hibernating in winter. Please, when you see a protein name in your exam or some chemical that you never heard of, do not panic, okay? Because I used to panic when I used to hear these weird chemicals or weird things or proteins. Don't panic. It's going to be related to what you studied. Just don't panic. Okay, so this protein is found on platelets and is involved in the binding of platelets to damaged blood vessels. Okay, so now we understand that this protein is found on platelets, meaning it's involved in the binding of platelets to damaged blood vessels, basically saying that this protein acts like a platelet and it basically makes a clot. So that's our basic understanding from the texts. Okay, discuss whether this discovery could be useful to humans. Okay, so this could be useful and not useful at the same time. So let's talk about it possibly being useful. So it would be useful if we can find a way to break down HSP47. Why, you may ask? Because reducing HSP47 would reduce the risk of blood clotting, because from this question we established that HSP47 basically makes blood clots, so reducing it will reduce the risk of blood clotting, therefore reducing the risk of coronary heart disease. And then you can also mention for another point, if you didn't mention any of these points, you can say that HSP47 may not even be present in humans, because you you can't link bears to humans and just say, oh, they have the same blood. Yes, they both are mammals, okay? Humans and bears are mammals. However, scientifically and chemically speaking, in our blood, it might not be the same. We have different antigens and stuff like that. Okay, so question 4. Obesity indicators are used to assess the risk of cardiovascular disease, CVD, okay, including coronary heart disease, CHD. Obesity indicators include body mass index, BMI, and waist to hip ratio, WHR. Describe the differences between CVD and CHD. Okay, so CVD includes all diseases affecting the heart and blood vessels, whereas coronary heart disease, okay, is when the coronary artery is blocked. Remember, the coronary artery is on the outside of the heart. So so this is our heart. Our coronary artery is on the outside of the heart. This is our coronary artery. And CVD is like deep vein thrombosis. Okay, so a woman who was 165 centimeters tall, had a mass of 68 kg, a waist circumference of 85 centimeters, and a hip measurement of 101 centimeters. Calculate the BMI of this woman. Well, that's very nice of them to give you the formula. So mass in kilograms, which is 68, over the height in meters. So I converted centimeters to meters by dividing by 100. And then remember the square root, which gave me 25. Now calculate the waist to hip ratio of this woman. So waist to hip ratio is going to be our waist circumference divided by the hip. So it's going to be 85 divided by 101, which gives us 0.84. Okay, so scientists collected data to evaluate these two obesity indicators for their effectiveness in predicting death from CVD and CHD. This study involved 9,206 adults aged from 20 to 69 years from Australian cities. This study started in 1989 when measurements for the obesity indicators were taken. Risk factors, including blood pressure and blood cholesterol levels, were measured. Information about other risks were recorded but not measured. By the year 2000, 473 of these adults had died. The data were compared with the causes of deaths. Explain why high blood pressure is a risk factor for CVD. Okay, well, high blood pressure causes damage to the endothelial lining of blood vessels. Over here, we can see the lining of blood vessels. If there's high blood pressure and it's going super fast, there's going to cause ruptures for the lining. Okay, this would cause inflammation. Okay, now this inflammation may result in a blood clot, which restricts the movement of blood, restricting the movement of oxygen. However, it's set we only need 2 points for this mark. Okay, some other factors were recorded as either being present or absent but were not measured. Name 2 other factors that might have been recorded but not measured. Well, that's a very easy question. Diet and family history. Okay, and over here they give us a bunch of graph. The graph shows the BMI waist to hip ratio of the men and women who died due to either CVD or CHD. Discuss the usefulness of these 2 obesity indicators in predicting death from CVD to CHD? Well, it's only useful in men above 24.9 to 29.9. However, graphs of waist-to-hip ratio are more useful than BMI, okay? Because the BMI, someone can have a really high BMI if they're very muscular because muscles are more dense and more heavier, or someone's bone density can be very high, so their BMI, like someone who has a higher bone density, if a doctor didn't see them and they looked at their BMI only, they would be like, oh wait, this person is unhealthy. However, they could not be unhealthy, they could be perfectly fine and healthy because their bones are just simply heavier. Okay. Also, these graphs are not useful as there are no error bars, which means that the results might not be statistically significant. Because if I have an error bar that's like this, and this error bar is like this, and this one is like this, they are all overlapping. So, which means that there's possibly no statistical significance. Okay, now this is the last question for question 4, and I'm going to be answering from question 5 to about question 7 or 8 and other parts. Thank you so much for listening, and comment down below if you need this past paper. Thank you.